All right, hopefully you've had a chance to try to answer this question on your own. So what is happening if this M protein gets mutated? Well, think about this in terms of your lungs, right? Your lungs and the cells in your lungs are constantly going through cell division. You get damage, you get things in the air that are getting into your lungs. Those cells are constantly going to be going through cell division because they are dying off and they need to be replaced with better, newer cells. Now, if this M protein or M factor is not functional, what happens is the cell in your lungs that is trying to go through cell division, it's reaching out around it and it's saying, okay, there's no cells here. I'm going to continue to go through cell division. But then it runs into a cell and it says, oh, hi, okay, I'm going to stop going through cell division. But the problem is, in order to stop going through cell division, it has to have this M factor. So it says, oh, hey, I'm going to stop going through cell division. Oh crap, I can't stop going through cell division. And so that cell continues to divide. And this is why things like smoking can tend to cause lung cancer. It's because the cells in your lungs are constantly having to be replaced, right? Now, in terms of external checkpoints, we also have this thing called anchorage dependence, which is basically when a cell needs a surface upon which it is going to divide, right? Now, these cells exist in our body normally, but there are some cells that don't exhibit anchorage dependence. So I want you to think for a second, okay, what types of cells would normally flow through our body that do not need to be anchored to something in order to go through cell division? You think you got it? Actually, your blood cells do not exhibit anchorage dependence, and that includes cells in your immune system. Now, the problem is in a cancerous cell, when a tumor begins to develop and these cells start to go through cell division without any restrictions, tumors and those cells can actually break off into the bloodstream. And this can be really dangerous, and it's called metastasis, right? So not all cells are going to do this, and not all tumors are going to do this. You can have a benign tumor that's not going to continue growing, and it is not going to metastasize. But a malignant tumor, or a tumor that is continuing to go through cell division, is going to potentially experience this phenomena called metastasis. And this is when a piece of the tumor breaks off in the bloodstream and then flows through the bloodstream and goes into a new region of the body. So you can have a tumor that starts in your heart and then breaks off into the bloodstream and then travels to your digestive system and into your pancreas, right? So now that we've gone through these external cell signals, I want you to think about this video that I showed earlier, and I want you to think, okay, what were these cells, these cancerous cells? What factor were we not seeing shown? What factor were these cells visibly ignoring? Take a second to think about that question. Did you get a chance to think about it? Well, you could make the argument that they are ignoring all of these things, right? Eventually, they could be exhibiting anchorage dependence, where they break off and flow into the bloodstream. You could be saying that growth factors are not being or are being received in high quantities. But the most direct answer is density-dependent inhibition or contact inhibition. These cells are not exhibiting contact inhibition. All right, so now we're going to talk about internal cell checkpoints, and these tend to be a little bit more difficult, so they may take a little bit more time. Internal cell checkpoints include P53 as well as other regulatory genes in your DNA. They also include these molecules and proteins called CDKs and cyclins, and they're also going to include molecular signals. So let's talk about P53. P53 is considered the guardian of the genome, and it is a gene associated with a protein that directly causes the suppression of tumors and damage to DNA. So let's take a look at this diagram that we have on the screen, all right? You get some sort of damage to your DNA, right? Maybe you smoke a cigarette and that causes damage to the cells in your lungs. Maybe you go out in the sun and you get radiation damage to your skin cells. But either way, you get some sort of DNA damage. Well, P53 is a protein that will run along the strands of DNA and say, okay, you're good, you're good, you're good. Oh no, you're not good. You got to stop. We need to stop the cell cycle because we do not want this error to be replicated. So this has to happen before S phase, right? P53 is going to initiate an inhibitor, and then this inhibitor is going to associate with CDKs and cyclins that pause the cell in G1 before S phase. So during the process of organelle replication, DNA is going to be checked by P53, and that is going to then regulate whether the cell continues through the cell cycle, right? And again, we're going to talk about CDKs and cyclins in a second. So this G1 checkpoint is where the cell is checking for nutrients. It's checking for the presence of growth factors. Are growth factors being received? If yes, then we can continue through the cell division process. And it's also checking for DNA damage. And if at any point P53 finds any of these things are not what they should be, it will send the cell into G0 or back into G1, depending on what needs to be changed. All right, so P53 is in this diagram here in the middle. And here is my question for you, again, relating to cancer. In almost 50% of cancers, and in fact, it's finding to be almost 50-50, like exactly 50-50, P53 is mutated. Based on this diagram, why would mutated P53 allow cancer cells to develop? Use the image to help you answer this question. Also, this term here, angiogenesis, it refers to blood vessel development. It refers typically to the growth of blood vessels into tumors. So take a second to think about this question. Did you figure it out? If P53 is mutated, because normally P53 causes the gene expression of proteins that inhibit blood vessel growth into tumors, it stops the cell cycle from continuing, it repairs DNA, but if it can't do any of that and the cell is too far gone, it'll initiate apoptosis or cell death. But if P53 is mutated, none of those processes can happen, right? Let's take a look at another diagram. See if you can find where P53 is on this diagram. Did you find it? P53 is going to be down here in the nucleus. Okay, that makes sense because P53 is going to be checking DNA. Now, if the DNA is so damaged that it cannot be repaired, then P53 is going to initiate a Bax intermediate, an MT intermediate, which will then initiate apoptosis. Cell's going to die. But if P53 runs along the DNA and it says, okay, we're good to go, we're good to go, we're good to go, let's keep going, then it's going to initiate P21, which will trigger these CDK cyclin complexes. And this is the next step in our understanding of internal cell checkpoints. CDKs and cyclins are enzymes and cofactors that are involved in cell division. So just a quick reminder of this image. A CDK is an enzyme. It's a cyclin-dependent kinase. So I encourage you to remember what a kinase is. It's a specific type of enzyme. And this particular kinase is going to be involved in prompting the cell cycle, continuing cell division. Cyclins are necessary cofactors that CDKs need in order to function properly. So if P53 finds damage to the DNA, it's going to trigger an inhibitor that prevents CDKs from functioning. So let's take a look at how these work. There are multiple checkpoints throughout the cell cycle. Specifically, there is one in G1, at the end of G2, and during the M phase. At the end of G1, it's going to be checking to make sure that DNA has been replicated appropriately. But the phases in the other, or the checkpoints in the other phases, they're going to have a little bit of a different function. And in order to understand those, we have to understand the function of cyclins and CDKs. So a CDK is considered a phosphorylating enzyme. This is the definition of a kinase. And it will not be activated until a cyclin cofactor has bonded to it. Now, once it bonds to the cyclin, it will attract a phosphate group into its active site, as well as another target enzyme. So this is the enzyme that's actually going to do the job that we care about, right? In this case, cell cycle progression. This target enzyme will then be phosphorylated by the CDK and then go on to perform whatever function it needs. Maybe that's breaking down the nucleus. Maybe that's causing spindle fiber attachment. Maybe that's causing DNA synthesis. Whatever it is, it's going to initiate and continue the process of cell cycle progression. Now, CDK is always in constant concentration inside the cell. And it's going to be waiting in the cell. It's going to be hanging out, sensing, probing to see if there's any cyclins available. Now, cyclins are not made available in the entirety of the cell cycle. They will be triggered after certain events. For example, this particular cyclin, there's a variety of different cyclins, but this particular cyclin that we're looking at is going to start increasing in concentration during the S phase. So this is an ion or some sort of cofactor that is going to increase in concentration starting in the S phase of interphase. Now, if the cell at any point senses, oh, there's not enough cyclin at this point, we're not going to continue through the rest of the cell cycle. We're going to go back into G0 and wait. We need to wait a little while. Now, if there is enough cyclin, the cyclin is going to bond to the CDK, and this is going to continue the cell cycle because these newly functional CDKs can now go on to activate their respective target enzymes. And they are going to do things like break down the nucleus and cause spindle fiber attachment and so on, right? After a certain point, namely mitosis is finished, the cyclins will be degraded and the process will start all over again, right? So this is how cyclins and CDKs function. Now, during the G2 checkpoint, this is going to be when the cyclins and CDKs are checking for appropriate cell size, as well as DNA replication. And then during the metaphase checkpoint, it's going to be checking for chromosome spindle attachment and just in general checking to make sure that the cell has completed its process of cell division. Each of these phases is going to have different CDKs. Okay. Now, the last question I have for us is relating to these CDKs and cyclins. So this is a graph showing the concentrations of different cyclins throughout the cell cycle because cells are going to have different concentrations depending on which part of the cell cycle they are in. Now, MPF is a cyclin-dependent kinase that needs a cyclin cofactor in order to break down the nucleus and promote the formation of microtubules. So based on this graph, which of these cyclins do you think MPF is most likely to need? Take a second to pause the video and think about it. Do you have a second to think about it? Well, here's a hint. MPF is short for mitosis promoting factor. So maybe that helps you out a little bit. In this case, the cyclin that this enzyme is going to need is going to be the M cyclin cofactor, because as M cyclin associates with MPF, it's going to cause breakdown of the nucleus and the formation of microtubules. But that doesn't occur until mitosis is able to occur. So that's the end of our lesson on checkpoints of the cell cycle. Hopefully that was helpful.